Hello, I'm Les Bennett, and I'm with the American Association of Pharmaceutical Scientists. It is my pleasure to host this special series of interviews with individuals who recognize for their innovations and contributions to the pharmaceutical sciences. Speaking on behalf of the AAPS Distance Learning Committee, one of our goals is to develop a series of videotapes in which we invite eminent pharmaceutical scientists to discuss their points of view and their own personal contributions to the events, issues, and challenges that have shaped the direction of the pharmaceutical sciences. We will also explore their perspectives on where the pharmaceutical sciences are headed in the future. Today, it is our pleasure to have the opportunity to visit with Professor Gordon Amidon of the University of Michigan. Welcome, Gordon.
Thank you, Les. Thank you.
Let's start off with probably, I think, what everyone recognizes as your seminal, outstanding contribution of the biopharmaceutics classification system. And give us a little history of how that has developed and the impact that it's having on the pharmaceutical sciences.
Yes, I think I would say the precipitating event, the initial event, was probably doing a sabbatical at the FDA in 1990. I went there when I knew the center director at the time from my NIH study section activities, and they invited me to be a visiting scientist at the FDA. So I went there. You know, most faculty go to Europe on vacation, but I went to the FDA did some real work at the FDA. But I could see that the struggle that the FDA was having with coming up with some dissolution guidance, because they were trying to write something that was encompassing, but it was the biopharmaceutical properties as we understand them today are clearly very different and very dependent on the drug. And so at that time, before I'd conceived of the BCS approach, I thought we know some drugs are easy and some are difficult, and so is there some way we can capture that in a categorization or compartmentalization? So that's what began the initial events. How to capture the differences in the properties that are essential to development of an oral drug product. And I would say it was another 1 to 2 years until I conceived where do we scientifically stand? You know, what principle are you going to use to try and categorize drugs and drug products, but particularly drug properties? And when I finally realized that you start with fixed first law, you know, and that became solubility permeability, fixed first law applied to a membrane, solubility dissolution rate, that connected the physical properties of the drug to the biopharmaceutical properties. And I think that's an essential connection which today permeates both new drug discovery and development, but also drug regulation. And I could see biopharmaceutical sciences really spans the whole time frame of a drug's development and then subsequent marketing and the regulatory standards that we set for drugs. So I would say it was that event. I mean, prior to that, of course, I had worked extensively in drug absorption and also pharmacokinetics. I would say probably my first love was the physical chemistry, physical pharmacy, and I would have to say that I think the biopharmaceutical classification system, while focused on the biopharmaceutical properties, came out of that physical training, because I think you need to know fixed first law, for example, before you can conceive of, you know, the biopharmaceutical classification system. And I could see, going back one step further in my career when I worked in industry for several years, this in the mid-'80s, that the biopharmaceutical properties of the drug, and this is now 20 plus years ago, were kind of a mystery. You know, you make up your formulation, you give it to animals, you give it to humans, and you hope for the best. You know, and there wasn't a connection between the pharmacokinetic side, drug metabolism side of a drug's properties, and the pharmaceutical formulation side. And it was probably my experience in industry where I could see the importance of that role, and yet there was no way of really capturing that in some scientific principles or scientific understanding I mean, it took me another 10 years or so to really kind of come to what the essence of that problem is. And I think having found it and its widespread acceptance, clearly I found the right basis. So and I think I'm really, I think that is the most important from the point of view of public pharmaceutical quality standards of anything that I've done.
Very good. I've always been impressed that this is a scientific basis for decreasing the regulatory burden.
Yes.
And the FDA says that they're a science-based organization, and I think this has been a significant contribution in terms of science-based making regulation more relevant. Can you comment on, how you think it affected the FDA in terms of their relationship with the BCS?
I think that the BCS within the FDA and the guidance that resulted from the work that we did in the mid-90s has won awards within the FDA. I think it's used as an example of how science can impact and improve regulatory standards within a regulatory organization. And I fundamentally believe that a regulatory agency should be doing research relating to regulatory standards. Now, that's perhaps controversial within the FDA and within the industry, but nevertheless, to develop new standards, you need evidence, and you need public evidence. And so one of the steps that I think led to a successful implementation of BCS within the FDA was a scientific advance that occurred in Sweden, which I recognized early on, which was an application of some of my early work from the late 70s and early 80s for determining permeability in humans. To make a regulatory change within the FDA, you need human data. I mean, you can't make a change in regulatory standards that are going to impact human products without a human database that's public and defendable. And so I realized we had to establish some human data. And so through the collaboration, through scientific contact collaboration, the development in Sweden, I was able to get the Swedish MPA, the Swedish Medical Products Agency, to say if the FDA will support this, we will support this, and that led to a large research effort, collaborative research effort between Michigan and Uppsala, in this case, that led to an essential database. I call it my $1 million plot. You've seen it many times. This is never going to be reproduced again because it would cost you $10 million to do it today, maybe more.
Yeah, you say 20 million.
Maybe I've said 20. Well, it's going to cost $500,000 per data point. We got 25 data points, so it's at least $12.5 million. Yeah. And now, of course, today, we have animal and in vitro systems that can do the majority of that work. And so today, versus, let's say, 20 years ago, I mean, I can say we can predict with a high degree of confidence, not perfect, but nothing is, the fraction absorbed. Not systemic availability, but fraction absorbed is the upper limit to systemic availability. So it's an essential piece of information for drug development. By knowing the fraction absorbed, you know whether you got a chance or not for development of an oral drug product. 20 years ago, it was a mystery. 20 years ago, it was a mystery. So I think that's, I really think it's advanced the role of pharmaceutical sciences and the basis for pharmaceutical sciences and even the credibility, if you will. We have scientific approaches now to drug development, systemic availability, controlled release, where the physical pharmacy and the biopharmacy and the pharmacokinetics are becoming increasingly predictable today. Now, I mean, as a kind of a side point, I mean, the development of software, for example, for predicting absorption today, I think, is highly refined. I mean, not perfect because it will never be perfect, probably, but that concept of drug absorption, systemic availability, the role of biopharmacy, I would probably say, having probably my industrial experience working at Merck and then at SmithKline for a few years before going back to Michigan, really made me realize that there's a big gap in, between the biological properties of a drug, pharmacological properties, and the physical properties, and there was nothing in between. And that's where biopharmaceutics, I think, started to germinate in the back of my mind.
So let's switch gears. I recall your presidency of AAPS as having two sort of seminal contributions, and I'd like to talk about those. One of them was the development of these international regulatory workshops. And let's give us a little history on that, and then I'll go to the other one, which is the strong interaction with Latin America. So let's talk about the regulatory workshops first.
Yeah, I would say the workshops, which I am very proud of, and they have continued. We continue to teach biopharmaceutics, biopharmaceutical principles, as well as the application, particularly to the standards for regulating pharmaceutical products worldwide. Has had an enormous impact on increasing, I think, the visibility of AAPS and the role that a professional society plays in evolving the science and the standards. Maybe just a tangent for a minute, but I would say the public vetting, if you will, of BCS. We had many workshops on BCS in the mid to late 90s. And I think that public vetting of BCS was essential to getting it accepted. I mean, we know it is, but that was an initiative with AAPS and with FIP. I want to say, importantly, probably the precipitating event for the workshops was when I had to spend 12 hours in a car in the worst fog I've ever seen, going like 200 miles in India with Hans Uniger. And that's right, yes, right, that's right. And we had to do something. I mean, we're in the car 12 hours together. So, I mean, you just can't, you could sleep, but I mean, you still can't sleep for 12 hours. So Hans and I started talking about, a whole variety of professional things. And we came up with the concept of a joint AAPS-FIP workshop. And we said, let's move ahead. Let's get AAPS and FIP. Hans would take it to FIP, and I would take it to AAPS. And we would propose to do a series of workshops with funding, joint funding from both organizations. And both organizations said yes, and that was the precipitating event. The fog was the word. You couldn't see the white line outside the car. I mean, it took us 12 hours, And then we missed our plane. And then we get to the hotel, and they want Hans and I to room together. Now, I'm an easy to get along with guy, but Hans, no way. He wouldn't room with me. So.
But let's go. I'd like to probe a little bit on the impact, because this was directed toward developing countries. And
yes.
Talk a little bit about what you think we've done worldwide in terms of these workshops.
Yeah, broadly, I think the essential regulatory point to start with is that the label on the product, the label, which is where most of the cost of a pharmaceutical product is, connecting that label to the physical product. And how do we do that? We want to ensure systemic availability. That's not going to change. However, is that the best test, systemic availability? And the biopharmaceutical classification system is developed on the basis that, depending on drug properties, we can set a better standard that is simpler and easier, more implementable, more regularly conducted. And I think that can have an enormous impact on developing markets where there's less technology for doing full-scale in vivo bioequivalent studies. Now, I'm not proposing and have never proposed that we change that standard. The question is, what's the best test? And what's the scientific basis for that best test? And that's what the BCS approach allows us to develop. And in the developing markets, excuse me, in the developing markets, you have you have local industries that are making pharmaceutical products that have never been tested in vivo. Maybe some have. This could include the innovator product, potentially, because an innovative product, while the clinical studies may have been done in U.S., Europe, with a product made in the United States or Europe, the product that's sold in Chile might be made in Colombia. And is that product connected to that clinical phase 3 product in a scientific way? That can be difficult to determine sometimes. We have many pharmaceutical products that are old and haven't really been tested for bioequivalence. And the developing of a dissolution standard, which today the WHO regulatory recommendations are probably the most advanced, and are standards that developing countries can follow to assure the quality of the product in their markets. And so I believe that the BCS approach and the drug dissolution approach can increase the assurance of quality products to the public throughout the world. And I believe ultimately that's our role as a pharmacist, as professionals, is providing a quality product to the consumer, to the patient. That's our most unique role. And now, in BCS, which has become worldwide, I think largely through the AAPS FIP workshops, has developed a international network, or I would say is developing an international network, because if you're a regulatory authority in Chile and you need to set guidelines, you're not going to come to me. You're going to come to your local experts. So in order to impact the guidelines and standards worldwide, you have to develop this international network. And I think the workshops have been principally, if not solely, responsible for that, and the implementation of the BCS guidelines, which are in various stages of implementation throughout the world, and particularly in Latin America, where I've probably given 8, 10, 12 different courses, different countries. The BCS is being implemented in many Latin American countries, and that's a great satisfaction to me to see research, I think, good research, that can, I think, significantly improve our confidence in the quality of products throughout the world.
So let's go back to that Latin America, because again, I view your presidency as something that said AAPS should concentrate and make an effort within Latin America.
Yes.
What was the driving force on that?
What was the driving force? I think that the part of it was BCS. I mean, I could see that it could have worldwide impact. And I could see AAPS is American. So let's be American. It's north, south, central. Let's be American. And AAPS had the resources and the scientific skill and the scientists like yourself that are willing to travel and promote, the biopharmaceutical sciences in Latin America. And I think it was a desire to do, to spread good science where it can have an impact on human health in the developing countries. And this is, we've also been in Asia, Southeast Asia. I mean, it's not like we've forgotten the other parts of the world. But I'd say the majority of my activities have been in Latin America, personal activities. And I believe America's American, north, south, central, and I think these workshops have certainly made AAPS far more visible. throughout the Americas.
Let's go back to your early academic career. I remember, of course, I'm significantly older than you.
Yeah, much older, yeah.
So I remember hearing about this phenom coming out of the University of Michigan, taking all these math courses, and I believe you got a master's in mathematics at Michigan.
Yes.
And tell, probably two things. Why are you in the pharmaceutical sciences? And what did you think this mathematics aspect of it was? Why was that so important?
Okay, good questions. I think I enjoy the applied sciences the most, no doubt. I mean, the application, seeing the application of the of good basic science that's impacting in pharmaceutical products. But the math, I think, goes back to my interest in understanding the physics and chemistry of a system. So as an undergraduate, a pharmacy student at the University of Buffalo, deciding on a research career, working in the labs, and taking physical chemistry, I went into the chemistry library to look at JPhys Chem. And so I started looking through this journal, and I saw all of this math, and I don't understand any of this. I said, I think to understand physical chemistry, you have to understand math. And so I decided, in addition to maybe having some intrinsic, which I still have today, interest in mathematical concepts, simple concepts like irrational numbers and, what is a circle? Some of these deep philosophical maybe questions about, what is math? Do we discover or do we invent math? Questions like that. That maybe attract some element, some philosophical element in me. But the practical implementation is you'd look at these papers and you see these triple integrals and stuff, and you say, what is this stuff? What does this really mean? What's the essence of this? And I decided that you had to take a lot of math. So I just said, okay, I'm going to take a lot of math. And then when I got to Michigan, I could see the opportunity to do kind of a joint program. And I just went after it, and I can remember sitting in a math class once, and this is probably what led to my lifelong interest, continued interest, even to continuing today, sitting in a math class in real analysis, which was a course I didn't want to take. I wanted to take applied math, partial differential equations and things like that. But, one of the requirements, what you take real analysis, and you have to get into things like, Lebesgue integrals and Riemann-Stieltjes integrals and things like that. But I can remember sitting in one class, and we were talking about, the subject was properties of real numbers. And after talking about the reals and the rationals and the irrationals and the transcendental and the algebraic numbers, all these different kinds of numbers with different properties, the math instructor asked a question which just floored me. And he said, how do we know all the numbers? I sat there, boy, I never thought about that. How do we know that we know everything about the numbers, or that we know all the numbers? Because we had real, rational, irrational, and then complex numbers. Maybe there's some more out there. How do we know? So I thought, boy, that's a really tough question. And I think for some reason that captures to me in my mind that there's some very fundamental, very deep principles within our conceptualization and development of mathematics in our culture that I guess I just have always been intrinsically attracted to it after that. I was attracted to it before, but I think I became more interested in it even after that.
Why are you a pharmaceutical scientist?
Because I was a stock boy in a pharmacy when I was in high school. I got a job. My uncle was a pharmacist. I think in our profession, it's such a small profession. I think many of our students, and certainly in my case, you learn about it through contact, through direct contact with a pharmacist or in a pharmacy. And when I looked at the curriculum as a high school student, I enjoyed math, science, chemistry, physics, math, but I didn't have a particular one that was favorite at the time. So when I looked at the pharmacy curriculum, it contained all of them, and I said, well, this isn't bad. And I was working in a pharmacy, and I saw being a pharmacist as something that I felt I could do. I said, I'm not sure what direction I want to take with my career. I mean, what do you know when you're 16 or 17? the pharmacy gave me multiple potential avenues to pursue. And I felt I could see myself being a pharmacist in small town Seneca Falls, New York. I mean, that was an acceptable career path for me, as well as maybe, I thought just maybe I could find some science, I could do some science, but I had no conception of graduate school and research at the time. But so for me, I saw a good professional career that was quite acceptable to me. I could see myself being a pharmacist, but I saw the opportunities to get involved in other things that were of interest to me, and I thought, well, I'll go and see. Go to college, take pharmacy, and see where I'll go with it. And I've never regretted for a moment that decision. Never.
And the scientist, what happened to you?
I think
to that pharmacist.
Okay. In my junior year, I decided to work in the labs, or I got a job working in the labs as a stock boy, and then I got some support to do research in the summers in the labs, and so I worked in the pharmaceutics and in the medicinal chemistry labs, and so I got to know the graduate students at the time, and became friends with the graduate students, and I could see one, myself doing the work, and I could see this is, I like doing this, and I like the people. And so it was the personal contact was a part of it, although the driver was, I like doing the work, the investigation, and that, doing something in the lab and then drawing your conclusions and implications from that, I guess I could see how a professional scientist could function. And I could see myself doing that, and seeing that I need to learn a lot to get there, but that I could envision it. So it was really working in the labs and seeing what a career a research and teaching career would be. I have to say I sought those out, to try and find out, get information, experience in order to make the decision. But it was readily available. You just had to seek it out, and I could see maybe it's partly personality. Why does one become a researcher versus a clinician? I think there's a certain you need to understand yourself in terms of what you find satisfying to do. When you go home at night. What gives you personal satisfaction? If it's a more clinical, day-to-day, moment-to-moment interaction with patients, if that satisfies you, you're probably oriented towards a clinical career. Whereas, my interest perhaps was more long-term, and that's a different personality, I think, or personality characteristic. And I have to say, the research career has just been, really completely satisfying to me.
Let's return back to that discussion of industry, academia, because you at one time actually held jobs simultaneously in both. And
yes.
Why don't you talk about the dynamics between them, the two forces, why you're sort of full-time academia now? Or?
Yeah, okay. I think that this is now the early to mid 80s, when I think biopharmaceutics was not a well-recognized component within industry. And I could see that while working in industry was a challenge and that I enjoyed. That is, it was, I enjoyed doing the science, I enjoyed the people, I enjoyed the challenges within industry. And I think the decision to go back into academics was, when I sat back and thought about it, was the ability, the graduate training and the research that you could do. And probably, there's somewhat more freedom within academics to pursue, research directions. But I could see the challenges within industry. And when I decided to go back to Michigan from Merck, there was another company, SmithKline, Beckman at the time, now GSK, that wanted me to start a drug delivery and biopharmaceutical effort within their company, and I decided that there's just no. I decided to go become academic, and I'm going to be academic. So I'm not going to go back into industry. Well, then they come back and said, well, how about if we have a joint appointment? So if we can't get you full time, we'll take you half time. And that was enough to get me interested because I liked the research. I mean, I felt the industrial application of pharmaceutical sciences, and particularly the biopharmaceutical sciences, was something I could impact in industry. And I enjoyed both arenas, going back and forth. So for 3 years, I would spend 1 week at Michigan and 1 week in Philadelphia at SmithKline Beckman, directing their drug delivery research. And I mean, I enjoyed both environments, and I think that's what ultimately really impacted my research in the biopharmaceutical area, seeing the importance of that role within the industry and the gap that existed between the physical pharmaceutical formulation, drug delivery development, and then the pharmacokinetic and pharmacodynamic sciences that really impact, of course, controlled drug efficacy. So connecting those two. And I enjoyed working in both areas. But it was very clear that I was ultimately an academic. And after 3 years, I'd have to say it wasn't 3 years of 2 half-time jobs. It was 3 years of 2 full-time jobs. I decided that was enough.
Let's pursue the academic. And you've trained many students and at the PhD level and also at the undergraduate level. But let's concentrate on the PhD students and how do you feel about that? I know you're very proud of the number of students who've gone into academia, but just I'd like to hear your just general impression.
Yeah, I am. And I mean, I think each student has to make their own decision. I mean, they have to decide what is personally, take ownership of their decision, and just like your children. But I think the way I teach and the way I interact with, I think the students come through the program with a real basis for scientific question and scientific thinking. And I'm very proud of the fact that more than 20 of my graduate and postgraduate students have chosen academic careers worldwide. And I think it's just that focus on science, whether it's an application, a pharmaceutical, biopharmaceutical application to pharmaceutical standards, or whether it's a basic transport mechanism or transporter or drug metabolism in the gastrointestinal tract impacting systemic availability. And I have group meetings where all of the students takes about 10 minutes to give an update on what they've done, whether they're doing dissolution work or whether they're trying to purify a protein. So we have a diverse, every 2 weeks, we have a diverse presentation of research. So the students get the training in this broad area. At least they may focus, let's say, on purifying a protein and identifying a metabolizing enzyme, but they're also listening to the students that are working on drug dissolution, pH, and whether bicarbonate is better than phosphate for a buffer. So they get that. They become aware of the diverse science that impacts biopharmaceutics and systemic availability of drugs. And so I think that breadth of exposure, and of course students get it in seminar in a pharmaceutics department, but I think it's that research interaction and that their colleague across the bench is doing something completely different, but yet focused on that biopharmaceutical science. I think to do research you have to have a broader view of science to see where science is going and what you would what you want to pursue, and it changes, of course, over time. So that broad view, I think, helps you decide what directions you want to go over the course of a career. So and it's probably partly laboratory ambience, that the fun of doing research. the enthusiasm that you bring in. So I think, I don't think I particularly encourage or academic careers because, I think the student has to make their own personal decision. But I think our laboratory environment encourages that. And so students, I'm sure, think about that significantly or periodically during the course of their career. And no matter what they do, I will help them try and find an appropriate position or recommend them. I have 3 students or 4 students doing postdocs now, 2 in Europe and 2 at NIH, and they'll undoubtedly be academics, take academic positions. Helping them find the right position, the right postdoctoral laboratory to advance their training. I like to see my students be successful, feel successful themselves, and help them find those right opportunities to get them the next step. And then, you're on your own, but yeah.
Should a young person in college today interested in the sciences consider the pharmaceutical sciences as a career, and why?
Oh, absolutely. Yeah, absolutely. I'm from the point of view of doing some science that provides a real benefit to patients, consumers, or the public. I think my view of an academic, I'm funded at a public, paid for at a public pie, the public, government, state, federal funding, but I'm paid for ultimately by the public. And I feel that in the role that in the pharmaceutical sciences, you can provide some payback. That is, you can do something. I'm not a purist, and I believe that you owe something back to the public. And the pharmaceutical sciences gives you an enormous range of opportunities to have your science provide something back that increases or improves the human condition. And maybe that's a little too broad, but that's how I feel. So that's I think that's really the essence of it. You can provide something back.
Can you give us a perspective of where the field's going? What do you think pharmaceutical sciences is moving toward, or where we're going, or how we're progressing?
Well, I think that we are entering a new era. When I look at where we were 15 years ago, we were working in the area of oral absorption. Just as I'm starting out with a specific example, and we could show competition, we could show carrier saturation, but we had no clue what the mechanism was. It was just an observation that we would do in our experimentation. And this is less than 15 years ago, that we had no idea of mechanism. And by the mid-90s, the biology of membrane transporters had developed to the point where it could actually be implemented in our laboratories through tissue culture techniques. All of this came out of probably the mega human genome effort and genomes of other species. I think that science will ultimately allow us to know more. In the next 50 years, we will learn more about human health and disease than we've learned throughout the entire history of human history. We're going to learn more in the next 50 years, maybe the next 25 years, about human health and disease than we've ever known. So that from the point of view of the potential for science to impact human health, it's just enormous. It's just enormous. Now, the pharmaceutical industry, while we have this other force with regard to pharmaceutical products and pharmaceutical quality, development of pharmaceutical quality, and the regulatory standards and the huge costs of drug development. Which has put our industry under huge pressure. But I believe that the science itself is just exploding, and so I see the potential in the future for pharmaceutical scientists and young pharmaceutical scientists to see in their lives, in their scientific lives over the next 30, 40 years, more advanced than we've ever seen before in human history. And so I think it's a tremendously exciting time to be in the pharmaceutical sciences, because we've got tools today that you couldn't even dream of 15 years ago.
Gordy, you've had an outstanding career, big impact, continuing to have big impact, and thank you so much for sharing.
Okay. Thank you, Les. It's been a pleasure. Thank you.